must announce that our speaker and I share a common educational background. We both did our undergraduate work at Chinese universities in Shanghai. He at what is now the leading school in that city, Fudan University. I many years earlier at the then leading institution, St. John's University. He has entitled his talk, The Genetic Basis of Evolution. This sweeping title permits him to talk about virtually anything. But I suspect we shall hear a sampling from the impressive array of research papers published since his graduation from Penn State in 1998. How anybody can graduate as late as 1998 seems incredible. Many of these studies investigate familiar evolutionary concepts using novel genomic information. Professor Zhang. Where is he? Here.
Thank you. How do I start? Okay, here we go. I want to first thank the organizing committee for inviting me, and I didn't realize how difficult the task is. I accepted the invitation without much thinking. Only a few weeks ago when I started preparing slides, oh my God, this is hard. Because as Doug said, he graduated from high school in 1959, so he witnessed all the evolutionary development in the past 50 years. It would take another 29 years for me to graduate from high school. Okay, so it was hard, but I tried. Okay, so the first question I had as a geneticist was why Darwin was wrong about genetics in his book and why he didn't know about genetics. Okay, of course, modern genetics started by Greg Mendel. Greg Mendel is a contemporary of Charles Darwin. Why Darwin didn't know about his work and didn't incorporate it into at least the later editions of the origin. So Greg Mendel started his experiment in 1856. That's 3 years before the publication of Origins. And he, from 56 to 63, he conducted a lot of experiments in pea plants. Okay, and he essentially finished the pea plant experiment, and he wrote his paper and read his paper in 1865. Okay, and published it in the following year. So the publication was 1866, but somehow it was not recognized by scientists at that time for the next 34 years. Okay, so why Darwin didn't know about Mendel's work? So first, Darwin didn't subscribe to the journal where the paper was published. Okay. And it's also believed that there were only 11 citations of Mendel's work before 1900 when Mendel's laws were rediscovered. But at least 3 of the 11 citations were accessible to Darwin. Okay, let's take a look at those 3. The first was in the Royal Society's Catalog of Scientific Papers, which was published in 1879, 3 years before Darwin's death. Okay. So as a fellow of the Royal Society, Darwin has had access to this book. However, this catalog did not have any indication of the content of Mendel's work. And as you see from the title of his paper, Experiments on Plant Hybridization, you have no idea what's in it, right? That's the first reference. The second reference was a book published in 1881, 1 year before Darwin's death, by Folke. The book is about plant hybridization. Very interestingly, this person referred to Mendel's work 18 times in the book. but he was mostly talking about not pea plant hybridization work, but other plants, okay? And in the most crucial part, page 108 to 110, in which reference is briefly made to the pea experiments. These pages remain uncut in Darwin's copy, so Darwin apparently didn't read, okay? And the third reference was also a book published by Hoffman in 1869, so this is quite early, 3 years after Mendel's publication. And Darwin apparently read this book quite carefully. He annotated this book. He made a lot of notes. And he even cited this book in his paper, The Effects of Cross and Self-Hybridization. But although Darwin cited this book, he didn't refer to Mendel's work. And also it's apparent that Hoffman, in his book, although he cited Mendel, he didn't realize the importance of Mendel's work in genetics. Okay, so given that Mendel was ignored by the entire scientific community for 34 years, I think it's fair to say that even if Darwin had studied Mendel's papers, he probably wouldn't realize the importance of that, particularly in relation to Darwin. Okay, that's all I found on the web. So I had a question, that is why Mendel didn't write an email or letter to Charles Darwin. And I couldn't find an answer to this. And so maybe historians in the audience can tell me. Because I would think that Mendel, after reading Darwin's book and knowing his own results, probably can make the connection and write a letter to Darwin. So I don't know why it didn't happen. So of course, the incorporation of genetics to Darwin's theory of natural selection happened in the first part of the 20th century, and the result of that is the Neo-Darwinism. So since Neo-Darwinism, which was well accepted by the end of 1950s, I think there are two major revolutions in terms of the genetic techniques and genetic data. Okay, the first is the molecular revolution, molecular genetic revolution. The second is the genomic revolution. So through the molecular genetic revolution, basically there are three things we can do. First, it provides a lot of molecular markers for studying evolutionary histories of species and populations. And the second, it provides the tools and the techniques for identifying the molecular genetic basis of phenotypic variation. And number three, it provides means to understand the mechanisms of evolution, particularly at the DNA sequence level. Okay, so that's molecular evolution. And the second phase is genomic evolution, which happened in the last 15 years or so. And genomic revolution provided more markers, molecular markers, and also new types of markers. For example, genomic rearrangement is a new type of marker, okay, for studying evolutionary history. And it provides also new mechanisms of evolution that were not known or were not well known in the molecular area. For example, horizontal gene transfer, okay. And also, genomic revolution provides the information for us to assess the relative contributions of various patterns or mechanisms of evolution. And throughout these 50 years, I think there's only one truly revolutionary thing conceptually in evolutionary biology. That's the neutral theory, like I already mentioned that. So what I'm going to do in the rest of my talk is to summarize what I think are some emerging rules from the studies of evolutionary genetics. Okay, so the number 10 is arbitrary. I picked 10 to fit the time frame for my talk. Okay. Also, this is a very personal view. Okay, I would like to hear the feedback from you. So the first rule, I think, is that life is fundamentally conserved. Okay, between very divergent species, even across the three domains of life, we see that the basic molecular cellular processes, signaling pathways, genetic code, and a lot of genes are shared. Okay? So from a molecular cellular biologist point of view, not much has changed since the last universal common ancestor of all existing organisms. From at the DNA level, we know that most of mutations are deleterious. So the dominant form of natural selection is purifying selection, removes most of the variations generated by mutation. Okay. And this extreme evolutionary conservation suggests to me that first, maybe there are only very few ways to construct life. Okay. Second, evolutionary contingencies were very important, probably because of those early events in evolution. The later evolution cannot create other forms. Okay. And I want to say that this extreme conservation is actually the foundation for model organism-based research. Okay. The second point I want to make is that chance plays a very important role in evolution. There are stochasticity at all levels. Okay, at the molecular level, for example, proteins interact with each other, but in a given cell at a given moment, whether they interact or not is by chance. Okay, and there are many other processes like transcriptional initiation. Okay, that is stochastic. Protein degradation is stochastic. There are many stochastic events: mutation, recombination, of course. And also at the organismal level, reproduction and survival is also stochastic. So chance plays a major role. From neutral theory's point of view, a large fraction of nucleotide substitutions in a genome are due to random fixations of neutral mutations. Also, if we compare species or populations of different size, population genetic theories tell us that chance is more important in smaller populations. So this has been the main argument behind Michael Lynch's hypothesis of the origin of genomic complexity. Basically, he's arguing that the genomic complexity originates through a passive process. It's because the population size gradually reduces, so many of the slightly deleterious mutations are acceptable. The third point is there's a lot of junk in the genome. So in contrast to the view that genome or organisms are perfectly adapted, we now know that there's a lot of junk in the genome. For example, in the human genome, there's only 1.5% that codes for proteins. Of course, there are some non-coding sequences that are important, but the estimate is that only about 5% of the genome is constrained, so are under the surveillance of natural selection. 95% of the genome are probably not functional. Okay. Of course, there's also the interesting observation. So people are very interested in identifying functional elements from the so-called junk part of the genome. Okay, and there's very interesting finding that is there are regions in the non-coding part of the genome that seems to have some molecular functions. For example, there's a piece of DNA that binds to protein. However, this piece of DNA is not conserved between species. So there's discordance between evolutionary conservation and molecular function. And what that means, I don't know. And the fourth point is there are a lot of horizontal gene transfers between species, at least in prokaryotes, okay? Between bacteria archaea, within archaea, but there are also horizontal gene transfers between prokaryotes and eukaryotes, although much less frequent. So when I first saw this picture, I saw, wow, Darwin already know horizontal gene transfer among branches of life. Of course, this is not horizontal gene transfer. He was using those horizontal lines to mean different times. Okay. So horizontal gene transfers are shown here. Those are the lines across different branches of life. I think this is a if you ask me what is the most novel findings from the genomic era for evolutionary biology, I think this is. Because this changed the way we think about evolution. So if there are lots of horizontal gene transfers between species, say 80% of genes are horizontally transferred between species, then what is the meaning of a species tree, for example? Okay, so it has very profound implications. The fifth point is that there is no clear relationship between gene number and organismal complexity. So when the human genome was sequenced, it was a surprise that we only have about 20,000 genes. So E. coli has 4,400 genes, yeast has 6,000, fruit fly has 13,000, nematode has almost same as human, 19,000. And then sea urchin, Arabidopsis, and rice, they all have more genes than we have. Okay, so apparently there is some mismatch between the organismal complexity and the gene number. Okay, so then it was found that there's a lot of alternative splicing in human, of course also in other high organisms. And alternative splicing increases the number of proteins in an organism. But it remains unclear how much or what fractions of alternatively spliced forms are actually useful, actually have distinct function and useful. This is unknown. In the past 5 years or so, people have started looking at the interactions between proteins and the genes in model organisms, and it is generally thought that those interactions are important. These are the sources of organismal complexity, but we don't really know whether they are or not. The sixth point is that gene and genome duplication is the primary source of new genes. The majority of the new genes that appeared in evolution came through duplication of pre-existing genes. Okay, so genome duplication is also quite frequent in plants, although they are much less frequent in animals, in fungi, and also in prokaryotes. After duplication, duplicate genes diverge. Okay, there have been several models to describe how they diverge in function. Okay, the first is so-called a neo-functionalization model proposed by Susumu Ono. Basically, it says after duplication, one copy would retain the original function, the other copy would acquire some new function. And a more recently proposed model is called the subfunctionalization model. It says that after duplication, the two daughter genes would each inherits part of the ancestral functions of the progenitor gene, and they diverge. So it's possible, and it's probably true that both models are correct. So some genes take this way, some genes take the other way, and also one gene family can use both. Okay, you can, and my own study shows that often after gene duplication, the early phase is subfunctionalization, and after subfunctionalization, the both copies would have to be retained in the genome. Then there's a long time for them to acquire new functions. We know that many evolutionary innovation requires or are due to duplications. Okay, examples are animal homeotic genes, vertebrate olfactory receptor genes. So olfactory receptor genes form a very large gene family. I think it's the biggest gene family in mammals, including human. There are hundreds to over 1,000 copies of olfactory receptor genes. The seventh point I want to make is that there are changes in protein function and changes in gene expression are both important in phenotypic evolution. This has been a subject of debate in the past few years. Okay, there are many examples where protein function change are important. There are also many examples in which gene expression change are important. I just give you one example, the alpha and beta globins that form hemoglobin. We humans have multiple copies of alpha globin genes and multiple copies of beta globin genes. And these different copies have different expression time. They are expressed at different stages throughout development. They also have different binding affinity to oxygen and CO2. So there are both changes in gene expression and changes in protein function. It has been proposed by Sean Carroll and others that the relative importance of the two types of genetic changes, expression change versus protein function change, may be different for different types of traits. So it's been proposed that expression change may be more important in the evolution of morphology, while protein function change may be more important for the evolution of physiology. And I have some unpublished data that actually supports this view. The third point is there are some in vitro evolution studies. Now we can now sequence the end product of experimental evolution of E. coli or yeast and actually look for the mutations that were responsible for adaptation. So I looked at the two recent references, and in both references, most of the adaptive mutations are either protein functional changes or copy number changes. Okay. Well, you can say copy number change is a type of gene expression change because the expression level changes, right? But I have to say that both of the experiments are physiological adaptation, not morphological change. Okay. The eighth point is that not all evolutionary changes are very small. Okay, so Darwin emphasized that evolution is gradual and by very small steps. But actually, from genetic studies, we know that not all are small. For example, some neutral or beneficial mutations grossly affect the genomic structure. For example, the genome duplication, of course, it's big, right? And segmental duplication, there are also large chromosome deletions, inversions, and translocations. Some evolutionary events have very big impacts, like endosymbiosis or horizontal gene transfers. Okay, there are some beneficial mutations that have simple mutations, like point mutations, that have very large phenotypic or fitness effect. So I got this table from a paper published just last week in Nature. This is the result. This is the analysis of the genome sequences for the E. coli strains that Rich Lansky was evolving in his lab for the last 20 years. Okay, so they sequenced the end product, and they found some mutations, and they measured what's the fitness contribution of those mutations. And you see that, for example, a mutation in this gene had a fitness advantage of 13.3%. It's a huge fitness advantage. You see that there are a number of mutations that have very large fitness effect. It is well recognized that abundant genetic polymorphisms exist in most species. So compared to the neo-Darwinism view, the molecular level polymorphism that we now know is very, very high. Okay? But it is actually consistent largely with the predictions of the neutral theory. Okay? So the neutral theory predicts that the nucleotide diversity is equal to 4n mu, and its effective population size mu is the mutation rate. And we now know that the maintenance of the polymorphism in populations is largely the balance of mutation, drift, and negative selection. It is very rare to observe over-dominant selection or balancing selection that maintains polymorphism. And also we now know that there are a lot of slightly deleterious mutations that are segregating in natural populations. Okay, finally, talking about speciation. So in the past decade or so, there have been some so-called speciation genes cloned. So these are genes that are incompatible between closely related species. Okay, they are responsible for reproductive isolation between species. And there are 11 of such genes cloned so far. If you look at the function of those genes, they're not concentrated in any one particular category, although there are a few DNA binding proteins, but there are also many other transmembrane growth factors, nuclear pore complex member, histone modifiers, many different forms. Also, if you look at whether those genes were under positive selection, there are about half of them that are under positive selection. All of them are from Drosophila. There are half of them that are not under positive selection. So given that positive selection is relatively common in Drosophila, it may not be special for these many speciation genes to be under positive selection. Okay, in the next few minutes, I want to talk briefly about some major questions I think can be solved in the next few decades. First, molecular genetic basis of macroevolutionary changes. I think now, at least in principle, it is routine to identify the genetic basis of phenotypic differences between closely related species or populations. At least we have all the methods available. It just takes some effort and time. Okay. But it is still very challenging to identify the genetic basis of phenotypic differences between very distant related species, such as between animal phyla. Okay. Because a lot of genetic tools are not available. Introgression, QTL mapping, association mapping, a lot of tools are not available. And also, even when you have a candidate gene, it is very difficult to rigorously test whether this gene indeed was the cause, okay, because of the big difference in genetic background. And I think a better understanding of developmental pathways and the gene-gene interactions in model organisms can help us. The second challenge is the molecular genetic architecture of multifactorial traits that vary between species or populations. And this is particularly so if the genes have relatively small contribution to the phenotype. Okay, we're talking about those quantitative traits. It is probably even more difficult to understand why these genes control this particular trait, even after you identify the gene. Okay. I think that in addition to QTL and association mapping, systematic gene deletion studies and systems biology can help us. Number 3 is epistasis. Epistasis means interactions between genes. Sometimes people say epistasis between sites, between amino acid sites within a protein. Okay. It is very important for many evolutionary theories. We all recognize that, but we try to pretend it doesn't exist because we don't have empirical data about epistasis. And in the past few years, systems biologists and functional genomics start generating gene-gene interaction and protein-protein interaction data for model organisms. I think in the next 5 to 10 years, we will have a good picture about epistasis. And the next is pleiotropy. It's also a very common phenomenon. It refers to the observation that one gene or one mutation affects multiple traits. Okay, and it's also very important for many evolutionary theories like cost of complexity, trade-off, aging, etc. But our empirical knowledge about pleiotropy is also very, very poor. Only in the past few years, through functional genomic studies, we begin to know the patterns of pleiotropy at genomic scale for very simple model organisms. So I think this will be a field that will have quick development by system biologists. So this is a long-standing question, the relative roles of selection and the drift in evolution. And if you ask 10 population genetics or evolution genetics, you will get 10 very different answers about this question. And I think we need a better definition of the question, and also better tools and a better and more data to address this question. And I think this can be very well answered in the next decade. And finally, there are some very well-known phenomena, evolutionary genetic phenomena, that still do not have clear answers. Okay. So, for example, codon usage bias, which has been studied for over 20 years. But we don't have a clear understanding of the cause of codon usage bias. Is it due to translational efficiency, translational accuracy, or both, or something else? There is no clear answer to this question. So I list some questions that I'm familiar with, but this is just a small sample. Another interesting observation is that there's a very, very strong negative correlation between the expression level of a gene and the evolutionary rate of this gene in terms of the protein evolution rate. There's a very strong correlation. R squared is like 0.3, 0.4. Okay. But there's no good explanation why gene expression level is such an important determinant of protein evolution rate. A recently discovered phenomenon is that in high organisms like human, there's a widespread transcription of non-coding regions. Okay? And whether those transcripts are functional or not is unclear. Because if you compare between species, those transcripts are not conserved. Okay, so you would think these are just noise. But on the other hand, if so much of the genome is transcribed, it's going to waste a lot of energy. Okay. So why there is no selection to prevent such thing? Finally, as I mentioned, the gene number and organismal complexity are mismatched. So what is the real genetic mechanism for organism complexity is an interesting question. Okay, with that, I would be happy to answer any question.